Hello students, welcome back to a new video of Sunanda's tutorial. So in this video we will discuss about the properties, structure, classification, types and function of lipids. So lipids comes under the unit 1, chapter 2, unit 1 of CSIR NET life science syllabus, and the unit 1 is structure and function of biomolecules, and the particular topic or the particular chapter under which that lipid comes is composition, structure and function of biomolecules. So today we will study about lipids. So these are group of diverse macromolecule consisting of fatty acids and their derivatives, and these are mainly insoluble in water, but it can be soluble in organic solvents. It consists of fats, oils, hormones and certain components of membrane which are grouped together because of their hydrophobic interactions. Okay, so they have water heating properties. These lipids are very essential constituent of diet because of their high energy value. Okay. These are also essential for fat soluble vitamins and the essential fatty acids found with the fat of natural foodstuffs. Fats combined with proteins, which are called as lipoproteins, are essential constituent of cell membrane and mitochondria of the cell. Lipids are naturally present in living beings like plant, animals, microbes, which forms various components like cell membrane, hormones, energy storage molecules. So this is some basics about lipids. Now properties. It can either liquid or non-crystalline solids at normal to room temperature. Pure fat and oils are colorless, odorless as well as tasteless. These are energy rich molecules. So these lipids are energy rich molecules because their long hydrocarbon chains contain many carbon and hydrogen bonds. Okay, CH bonds. When these bonds break during cellular respiration, they release more than twice as much energy as carbohydrate or proteins release. So in this way, these are energy rich organic molecules. These are insoluble in water but soluble in alcohol, chloroform, acetone, benzene like organic solvents. They do not have any charge, ionic charge. Solid triglycerols or fats have high proportion of saturated fatty acid, and liquid triglycerols, oils, have high proportion of unsaturated fatty acid. What is saturated and what is unsaturated we will discuss later on. So now hydrolysis of glycerols. Glycerols, like any other esters, react with water to form their carboxylic acid and alcohol in a process called hydrolysis. Then saponification, it is another property. Triglycerols may be hydrolyzed by several procedures, most common of which utilizes alkali or enzymes called lipases. Alkaline hydrolysis is termed as saponification because one of the products of this hydrolysis is soap, generally sodium or potassium salt of fatty acid. Hydrogenation, the carbon-carbon double bonds in unsaturated fatty acids can be hydrogenated by reacting with hydrogen to produce saturated fatty acid. Okay. Then halogenation, unsaturated fatty acid, whether they are free or combined as esters in fat or oil, react with halogens by addition at double bonds. The reaction results in decolorization of halogen solution. Do you know what is halogens? Halogens are group of 6 non-metallic elements present in group 17 of the periodic table. Examples: fluorine, chlorine, bromine, iodine, etc. Okay. Next, rancidity. So rancid term is applied to fat and oil that develops unpleasant odor. Hydrolysis as well as oxidation reaction are responsible for causing rancidity. Oxidative rancidity occurs in triglycerols containing unsaturated fatty acids. Okay. Next, structure. So the main building block of lipids are fatty acids, which can be saturated or unsaturated, and glycerol. So unlike proteins or nucleic acids, they are not large polymers made up of repeating monomer. Instead, their chemical structure varies widely depending on specific classes. And the main building blocks are fatty acids and glycerol. Fatty acid can be saturated, containing only single carbon-carbon bond, forming straight chain that pack tight together, and unsaturated means contain one or more double bonds, creating links in the chain. Okay. Next, so these are the structure: saturated fatty acid, unsaturated fatty acid containing double bonds. Okay. Then classification. Lipids can be broadly divided into three groups: simple lipids, compound lipid, which are linked with other, like with phosphate, with carbohydrates to form glycolipids, phospholipids. Then derived lipids. Derived lipids are the components or derived components of simple and compound lipids. Okay. Then what is alcohol and what is esters? Alcohol and esters are the term which we will frequently use during the study of lipids. So glycerol make up simple lipid, which are esters of fatty acid and glycerol, and similar alcohol. So alcohol might be glycerol or other long chain alcohol. Long chain alcohol are mostly monohydroxy with single hydroxyl group, OH group. Depending on the alcohol use, simple lipid consist of fat or oil or waxes. Fats and oils are esters of fatty acid and glycerol, whereas waxes are esters of fatty acid and long chain alcohol. Okay. So triglycerides or triglycerols, these are most common and important type of lipid, which is the ester of 3 fatty acids and 1 glycerol. Triglycerides are the main constituent of our body fat, and it is also present in other vertebrates as well as vegetables. Structures: triglycerides are triesters where 3 fatty acids molecules are bound to a single glycerol molecule by covalent ester bonds. Okay. So now what is fatty acids? These are organic molecule of long chain carboxylic acids with 4 to 36 carbon atoms. The hydrocarbon chains are either saturated or unsaturated depending on the bonds between the carbon atoms. If carbon-carbon bonds are single, it is saturated, and if one or more carbon-carbon double bonds are present, it is unsaturated. Naturally occurring fatty acids are mostly unbranched, and these occur in 3 main classes of lipid: triglycerides, phospholipids, and cholesterol esters. Okay, so unbranched fatty acids are mostly present in cholesterol esters, triglycerides, and phospholipids. Okay, then functions of lipids. So these are chemically diverse group of compounds, and the biological function of lipids are diverse as their chemistry. In our body, fats serve as efficient source of energy, are also stored in adipose tissues. These also serve as insulating material in subcutaneous tissue around certain organs to protect those organs. phospholipids and sterols are major structural elements of biological membranes. Similarly, fats combined with proteins are important constituent of cell membrane and mitochondria of the cell. Okay. Lipids are also structural components of the cell and provide hydrophobic barrier that allows separation of aqueous content of cell and subcellular structures. Other lipids, although present in relatively small quantities, plays crucial role as enzyme cofactors, electron carriers, light absorbing pigments and hydrophobic anchors of proteins. Okay. Cholesterol acts as precursor of fat-soluble vitamins like vitamin D, hormones. Lipids are also activators of enzymes like glucose 6-phosphate, beta-hydroxybutyric dehydrogenase and steryl coenzyme A desaturase. Okay. Now, what is lipid wraps? These are specialized patch present in cell membrane. They are packed with cholesterol and fat called sphingolipids. These areas stay more solid than rest of the membrane. They act like floating work platform and help the cells to send signal and in the movement of proteins. Key features: these are made up of cholesterol and sphingolipids. Thicker than normal membrane, always moving, hard to dissolve by any other chemicals. Main function: they help in cell signaling, protein sorting and pathogen entry. Some virus bacteria use wraps to break into and infect the cell. They help to carry specific protein to carrier correct part of the cell. In this way they help in protein sorting and cell signaling. They group receptors together so that cell can react quickly to outside messenger or stimuli. So this is about lipid wraps and lipids. So with this I am concluding my lecture video here on the topic lipids, their structure, their types, their functions. So I hope I have touched each and every point, important points. So, and this video will be beneficial for you in future. If you liked it, please hit the like button, share with your other friends and please subscribe to my channel Sunanda's tutorial to get more important valuable videos like this. Thank you.